Welcome to this explainer on common honeybee pests and diseases. If you're following along with Blackwater Honey's Is Beekeeping For You series, you are in the exact right place. Today, we're going to equip you with a really calm, methodical, and visual triage system to monitor your hives. Our goal isn't to make you an instant veterinarian, but to give you the confidence to easily distinguish normal, everyday hive activity from actual red flag warning signs. So let's dive right in. Now, you might be surprised to learn that a perfectly healthy hive isn't going to look completely uniform. You're going to see gaps, and you're definitely going to see some oddities. But certain patterns absolutely demand your immediate attention. Things like the small hive beetle or a worker bee showing signs of deformed wing virus are clear indicators that something is off. But remember, we never ever diagnose a whole colony based on a single photograph or just one unusual cell. Here is our roadmap for today. We'll cover normal variation versus actual disease, the beekeeping triage system, common pests and intruders, brood and adult bee diseases, and finally, biosecurity and gathering evidence. Starting us off, Section 1, normal variation or disease. So before we hit the panic button and assume the worst, we really need to recognize what a baseline, healthy colony actually looks like. A lot of strange-looking things are completely normal. Seeing a few open cells in your capped brood, that could just be normal emergence or the queen's laying sequence. Finding pale, kind of fuzzy workers, those are just newly emerged bees, and they are completely healthy. Even brown or reddish propolis is just plant resin used to seal gaps, not some scary disease scale. And empty queen cups or busy circular orientation flights, just standard bee behavior. Context is everything here. But what happens when you see something that isn't on that normal list? Let's move to Section 2, the beekeeping triage system. When things look off, you need a reliable protocol to avoid guesswork. This 7-step clinical triage sequence is your safety net. It starts with observation and comparison, really looking at both sides of the frame. But step 3 is where a lot of beginners trip up. Contain. You have to stop swapping frames or tools immediately. Then you document with photos, confirm with a state inspector or lab, respond according to official guidelines, and finally, recheck. Having this system empowers you to safely handle suspect material without prematurely diagnosing anything or prescribing off-label treatments. All right, Section 3, common pests and intruders. It's super important to differentiate the physical intruders that can really wreak havoc on a vulnerable colony. And there's a mix-up that happens literally all the time between small hive beetle larvae and wax moth larvae. Small hive beetle larvae are firm, cream-colored, and they only have 3 pairs of legs up near the head. They're the ones that cause that terrible fermenting slime. Wax moth larvae, on the other hand, are softer, kind of caterpillar-like, with abdominal prolegs. They tunnel right through the comb, leaving behind silk webbing and destroying unprotected stored comb. Knowing the difference totally changes how you manage your equipment. Of course, beyond beetles and moths, your hive is basically an all-you-can-eat buffet for wildlife. You've got to protect your equipment from mice by popping in mouse guards before the cool seasons hit. And you'll want to deter larger predators like skunks, raccoons, and especially bears with proper elevation or sturdy electric fencing. Oh, and remember, while ants are scavengers that can stress a weak colony, native wasps are often really beneficial predators. Make sure you correctly identify them before you try to implement any control measures. We also have to keep our eyes peeled for broader environmental threats. As of August 2026, we are actively tracking the invasive yellow-legged hornet in Georgia, South Carolina, and North Carolina. We're also on high alert for Tropolelaps mites, a really fast-moving mite that is an immediately reportable suspicion, even though it hasn't been detected in U.S. hives yet. If you suspect either of these, safely photograph them and report them through official channels. And please, no matter what, never try to disturb a hornet nest yourself. Moving right along, Section 4, brooded and adult bee diseases. Okay, we have to talk about the one that keeps beekeepers up at night, American Foulbrood or AFB. This is a highly infectious, persistent, spore-forming bacterium that kills older, sealed larvae. It leaves behind a mottled, dark, sunken, and often perforated capping. If you suspect AFB, it demands the immediate closure of the hive and a call to your state apiary inspector. And here's a massive rule for you. Never use the matchstick ropiness test on every single suspicious cell. Doing that is a surefire way to spread those highly persistent spores across your comb and your tools. So, how does AFB compare to its similarly named counterpart, European Foulbrood, or EFB? Well, by comparing the older, sunken sealed larvae of AFB to the younger, twisted, melted-looking, uncapped larvae of EFB, we can gather the right evidence for the diagnostic lab. While AFB requires absolutely urgent containment, EFB also requires lab confirmation and careful management, since it's heavily influenced by colony nutrition and stress. Keep in mind, environmental stressors can cause other overlapping conditions that actually kind of mimic Foulbrood. Fungal Chalkbrood turns larvae into hard white or gray mummies. The viral Sacbrood creates these weird fluid-filled sacs. Or, honestly, it might simply be chilled brood found on the nest edges after a cold snap, or starved brood from a severe food shortage. Always look at the overall pattern and the recent weather history before assuming the absolute worst. Now, what about the adult bees? You might be wondering, how does varroa mite management relate to adult bee viruses? Well, controlling varroa is your absolute primary defense against adult bee illnesses like deformed wing virus and various paralysis viruses. The mites basically act as amplifiers for these pathogens. And speaking of adult ailments, gut diseases like Nosema require lab microscopy to diagnose. Seriously, do not try to diagnose Nosema based on simple fecal spotting alone. Spotting can just be from winter confinement or poor food. Sometimes, though, the threat isn't a disease at all. If you observe rapid, unexplained adult mortality, like, say, a sudden pile of dead or twitching bees out front, that really hints at pesticide exposure. Do not guess the chemical cause. Your job here is to strictly document the entrance, photograph any nearby blooms, and preserve samples exactly as directed by investigating state authorities. Which brings us to our final section, Section 5, Biosecurity and Gathering Evidence. Listen, strong biosecurity is absolutely your best friend. This means never feeding your bees unknown honey. It means stopping bees from robbing dead-outs or wet supers where microscopic spores might be hiding. You must strictly quarantine new colonies and used equipment. And crucially, use diagnosis-specific tool disinfection when disease is suspected. And please, I know it's tempting, but do not move suspect spotty brood into a strong colony to try and save it. You are highly likely to just infect your strongest hive. When you do need to call in an inspector, gathering the right evidence methodically is key. Step 1. Photograph the whole colony entrance and debris. Step 2. Take straight-on photos of the whole frame, both sides. Next, grab those clear macro close-ups of the capped brood, larvae, and pests. Finally, jot down sensory notes, things a camera just can't capture, like a sour odor, the texture of the comb, or the recent weather. And this brings us to our golden rule of thumb. Your ultimate responsibility as a beekeeper is recognition and reporting. Never prescribe unconfirmed or off-label treatments, antibiotics, or equipment destruction based on a single photograph or a single suspect cell. You absolutely must secure official confirmation, utilize veterinary oversight where applicable, and always follow state law and product labels. So, remember, observe the broader pattern, compare your neighboring frames, and contain any suspect material. Strong biosecurity protects every single colony in your yard. But keep in mind, not every unusual pattern means disease. Sometimes a thriving, overflowing hive is just getting ready to reproduce, which leads us to ask our next big question: is your colony preparing to swarm?